Welcome all to this new edition of the EMEUNET paper of the month video interview. My name is Javier Rodríguez Carrio, and I'm a member of the EMEUNET-EULAR Journal Club team. Today, we are going to discuss a paper recently published in Lancet Rheumatology from a group of colleagues from the University College London. So now let me introduce the first authors of the paper, George Robinson, postdoctoral researcher, and Junjie Peng, PhD students, both coming from the Center for Rheumatology Research and the Center for Adolescent Rheumatology versus Arthritis at the Department of Medicine, University College London at the UK. So welcome, George and Junjie. Your paper has been selected for this Paper of the Month edition. So first of all, congratulations and thank you very much for your participation and for sharing your time and your results with us. So first, can you please introduce the title of your paper?
Yes, I'll do that. So our paper is disease-associated and patient-specific immune self-signatures in juvenile onset systemic lupus erythematosus, patient stratification using a machine learning approach.
Okay, thank you very much. So I think a major breakthrough of your study seems to be the use of a machine learning approach methodology. So to start, can you please explain in simple words, what does it mean and why did you choose this approach? And more importantly, why this approach is important from the rheumatology perspective?
Yeah, so I think machine learning is a promising and exciting tool that has greatly influenced the medical research in the past decade. And so like machine learning is basically computer algorithm that can improve automatically through learning from the existing data. And so nowadays research with the advanced multi-omics technologies, we are able to generate a lot of metadata from the patients. And this data usually is large in size and has a high dimensions, which means there's a large number of variables. And sometimes this can be difficult for traditional research to select essential biomarkers for the research. And so machine learning can be quite powerful to process this kind of data and perform essential disease-associated and patient-specific immune characteristic in rare disease populations, such as GSLE. And in our pipeline of our study, we apply a variety of variable selections methods, including balanced random forest model, partially square discrimination analysis, logistic regressions, and multiple t-tests, etc. And so by using this model, we try to analyze the disease in a more comprehensive and productive way. And for the rheumatology study, I think one of the challenges is that many of the autoimmune rheumatic disease is highly complex and heterogeneous, both in the molecular levels and in the disease manifestations. And this makes the applications of precision medicine especially important, as patients could be quite different from one another and they're very likely to have response to treatment differently. And so I believe machine learning can definitely help a lot by identifying biomarkers and performing patient stratifications so that the treatment options for some patients can be more effective.
Okay, so thank you very much. It actually seems like a very complex methodological approach for a very complex disease. So it seems like we are fitting both aspects. So now let's go to the research question. So what was the main research question that you tried to address in your study?
So I'd say that the key question here was, can we identify key immune subsets involved in the pathogenesis of juvenile lupus using these modern machine learning techniques? And then secondary to that was to ask if we could use these key immune subsets to define subgroups of patients that have different clinical outcomes in the hope that we can advise personalized therapeutic approaches, which is becoming a more promising aspect of the current treatment options for patients.
Okay, Thank you very much. So now that we are talking about the diseases, a clinical question: why did your study focus on juvenile SLE and not in adult patients?
So the juvenile onset patients, it forms a very important group of lupus patients, as patients tend to have a more severe disease outcome clinically when the disease begins before the age of 18. But despite this, research into juvenile onset SLE is under-researched, but with the amazing platform of the Adolescent Centre versus Arthritis at University College London, we aim to change this and improve patient outcomes, particularly for these younger patients.
Okay, so now with methodology and the research question and the clinical picture, let's go into the results. So can you please summarize, although maybe it's difficult, in simple words, the main findings of your work for the broad audience?
Yeah, so this study is the first to report an in-depth analysis of immune cell phenotype in patients with juvenile onset lupus. And so by using the immunophenotype data from a total of 67 JSLE patients and 39 healthy controls, our machine learning classification model is able to identify patients with the juvenile lupus from the healthy control with overall 91% prediction accuracy. And from our analysis, we selected A immune cell types as the disease-associated biomarkers. And we then use these biomarkers, we are able to stratify the patients into 4 groups with distinct immunological profile. And finally, our trajectory analysis shows that these groupings are associated with disease activity and treatment outcome.
Okay, so coming back to this extensive characterization of immunological parameters, can you summarize all the features, all the immunological cells and all the immunological populations that you have included in your analysis?
So for the machine learning, we tried to cover a broad spectrum of the immune architecture that's measurable from the isolated peripheral blood mononuclear cells that we use. So we measure these by flow cytometry, and they include subsets of the innate response, such as monocytes and PDCs, but we also include cells of the adapted response, such as T cells and B cell subsets, and we go reasonably extensively into the subsets of these cells. But because all of these cells communicate with each other, we also used correlation analysis to find altered relationships in JSLE compared to healthy controls, as well as network analysis, which we use to find relationships between the immune subsets and commonly measured clinical features to try and make this more translational.
Okay, thank you very much. So now coming after this deep immunophenotype, did you was able to discriminate the controls and the patients using this immunophenotype, and were all the immune cell populations informing the same, or were any difference across these immune populations?
Yeah, so in our study, by using the immunophenotype data and one of our model, which is the balanced random forest classification model, is able to achieve an overall 91% prediction accuracy with 90% sensitivity and 82% specificity when discriminating between the patients and control. And I think this is overall quite good model to showing that we are able to separate the groups. And so by looking at the contribution of each immune cells in the model, we can then find out that the top immune cells that is driving the separations between the patients and the controls are mostly T cells populations, especially CD8 T cells. And there's also some of the B cells. And so I think these results suggest that the CD8 T cells might have an important role in the JSLE pathogenesis.
Okay, so it seems that there is not a profound or a general activation of the whole immunophenotype, but there are differences across the immune population. So what's the global message about the underlying pathogenic mechanisms? So is there a new novel pathogenic mechanism that you have maybe uncover or you can suggest, or on the contrary, are there different mechanisms in different patients? What do you think?
So as Junjie mentioned, we identified key subsets. There was 8 immune subsets that we found associated with the pathogenesis of JSLE, and validated these using different statistical methods. Each of these subtypes we hope will spark further research into pathogenic mechanisms. But going forward, we have a clear understanding that CD8 positive T cells could play a significant role in disease, where the effector memory subset appear to drive a more severe clinical outcome. So targeting these therapeutically could hold promise for specific patients. But a lot of our work going forward will be more functional, trying to identify mechanisms that may be affected between the immune cells, specifically in their interactions.
Okay. So, and also your study, apart from this discrimination and this understanding of the immune circuits, also looks at some associations with the disease trajectory, so with the response of treatment, with treatment outcomes, and also with disease progression. So what have you observed with these biomarkers, with this immunophenotype?
Yeah, so when we look at the 5-year longitudinal data of the stratified patients groups, we found out that the JSLE patient groups with the elevated CD8 effector memory T cells frequency, they had a more persistently activated disease over the time. And this can be observed by the SLE disease activity index score, as well as the complement-free level, which is another indicator for lupus activity. And also these patient groups with high disease activity, they also tend to have a higher treatment intensity. And this also reflects the low treatment efficiency in these patients and adjusts the importance of having a more precise and effective treatment options for them.
Okay, thank you very much. So I think we are coming to an end, so let's make the final question. So now, what can be the next step, the perspectives for your study, and how can we translate these findings into clinical practice? What do you think is the main application for them?
So as with all research, validation is key. So we hope that through collaboration, we can validate these results in other cohorts to build more supporting evidence of a key role of these specific immune subsets. And we can also use retrospective clinical data to monitor patients that have had a more complicated disease course, and then track back and measure their immune profile to again build further evidence of our signature for JSLE. But more functional experiments will be required from us going forward to identify pathways we can target therapeutically. But of course, the kind of downstream aim will be to try and get this into a clinical setting whereby you can treat to target specific immune cell types depending on patients' clinical feature at the time. So yeah.
Okay, so we are now at the end of this video interview. So George, Junjie, thank you very much for your participations. Thank you very much for sharing your results and your time with us. We will also like to congratulate the rest of the team at the University College London and all the authors for participating in this study and winning this paper of the Mount Paul to all this EMEUNET audience. So thank you very much again, and thank you very much from the EULAR and the EMEUNET Journal Club community. So stay tuned to our platforms for future video interviews. Thank you very much.
Thank you. Thank you very much.
Thank you, Javier.